Chapter 27 Introduction to Animal Diversity 27.1 Features of the Animal Kingdom. By the end of this section, you will be able to do the following. List the features that distinguish the kingdom Animalia from other kingdoms. Explain the processes of animal reproduction and embryonic development. Describe the roles that Hox genes play in development. Two different groups within the domain Eukaryota have produced complex multicellular organisms. The plants arose within the Archaeplastida, whereas the animals, and their close relatives, the fungi, arose within the Opisthokonta. However, plants and animals not only have different lifestyles, they also have. Figure 27.1 The leaf chameleon, Bruxia micra, was discovered in northern Madagascar in 2012. At just over 1 inch long, it is the smallest known chameleon. Credit. Modification of work by Frank Glaw, et al. PLOS Introduction. Animal evolution began in the ocean over 600 million years ago with tiny creatures that probably do not resemble any living organism today. Since then, animals have evolved into a highly diverse kingdom. Although over 1 million extant, currently living, species of animals have been identified, scientists are continually discovering more species as they explore ecosystems around the world. The number of extant species is estimated to be between 3 and 30 million. But what is an animal? While we can easily identify dogs, birds, fish, spiders, and worms as animals, other organisms, such as corals and sponges, are not as easy to classify. Animals vary in complexity, from sea sponges to crickets to chimpanzees, and scientists are faced with the difficult task of classifying them within a unified system. They must identify traits that are common to all animals as well as traits that can be used to distinguish among related groups of animals. The animal classification system characterizes animals based on their anatomy, morphology, evolutionary history, features of embryological development, and genetic makeup. This classification scheme is constantly developing as new information about species arises. Understanding and classifying the great variety of living species help us better understand how to conserve the diversity of life on Earth. Chapter Outline 27.1 Features of the Animal Kingdom 27.2 Features used to classify animals 27.3 Animal phylogeny 27.4 The evolutionary history of the animal kingdom. Different cellular histories as eukaryotes. The Opisthokonts share the possession of a single posterior flagellum in flagellated cells, e.g., sperm cells. Most animals also share other features that distinguish them from organisms in other kingdoms. All animals require a source of food and are therefore heterotrophic, ingesting other living or dead organisms. This feature distinguishes them from autotrophic organisms, such as most plants, which synthesize their own nutrients through photosynthesis. As heterotrophs, animals may be carnivores, herbivores, omnivores, or parasites. Figure 27.2a, b. As with plants, almost all animals have a complex tissue structure with differentiated and specialized tissues. The necessity to collect food has made most animals motile, at least during certain life stages. The typical life cycle in animals is diplontic, like you. The diploid state is multicellular, whereas the haploid state is gametic, such as sperm or egg. We should note that the alternation of generations characteristic of the land plants is typically not found in animals. In animals whose life histories include several to multiple body forms, e.g., insect larvae or the medusae of some cnidarians, all body forms are diploid. Animal embryos pass through a series of developmental stages that establish a determined and fixed body plan. The body plan refers to the morphology of an animal, determined by developmental cues. Figure 27.2 Heterotrophy. All animals are heterotrophs and thus derive energy from a variety of food sources. The black bear is an omnivore, eating both plants and animals. The heartworm Dirofilaria amidus is a parasite that derives energy from its hosts. It spends its larval stage in mosquitoes and its adult stage infesting the heart of dogs and other mammals, as shown here. Credit A. Modification of work by USDA Forest Service. Credit B. Modification of work by Clyde Robinson. Complex tissue structure. Many of the specialized tissues of animals are associated with the requirements and hazards of seeking and processing food. This explains why animals typically have evolved special structures associated with specific methods of food capture and complex digestive systems supported by accessory organs. Sensory structures help animals navigate their environment, detect food sources, and avoid becoming a food source for other animals. Movement is driven by muscle tissue attached to supportive structures like bone or chitin, and is coordinated by neural communication. Animal cells may also have unique structures for intercellular communication, such as gap junctions. The evolution of nerve tissues and muscle tissues has resulted in animals' unique ability to rapidly sense and respond to changes in their environment. This allows animals to survive in environments where they must compete with other species to meet their nutritional demands. The tissues of animals differ from those of the other major multicellular eukaryotes, plants and fungi, because their cells don't have cell walls. However, cells of animal tissues may be embedded in an extracellular matrix, e.g., mature bone cells reside within a mineralized organic matrix secreted by the cells. In vertebrates, bone tissue is a type of connective tissue that supports the entire body structure. The complex bodies and activities of vertebrates demand such supportive tissues. Epithelial tissues cover and protect both external and internal body surfaces, and may also have secretory functions. Epithelial tissues include the epidermis of the integument, the lining of the digestive tract and trachea, as well as the layers of cells that make up the ducts of the liver and glands of advanced animals, for example. The different types of 700 Chapter 27, Introduction to Animal Diversity, access for free at openstax.org. Tissues in true animals are responsible for carrying out specific functions for the organism. This differentiation and specialization of tissues is part of what allows for such incredible animal diversity. Just as there are multiple ways to be a eukaryote, there are multiple ways to be a multicellular animal. The animal kingdom is currently divided into 5 monophyletic clades: parazoa or porifera, sponges; pachyzoa, tiny parasitic creatures that resemble multicellular amoebae; cnidaria, jellyfish and their relatives; ctenophora, the comb jellies; and bilateria, all other animals. The pachyzoa, flat animal, and parazoa, beside animal, do not have specialized tissues derived from germ layers of the embryo, although they do possess specialized cells that act functionally like tissues. The pachyzoa have only 4 cell types, while the sponges have nearly 24. The 3 other clades do include animals with specialized tissues derived from the germ layers of the embryo. In spite of their superficial similarity to cnidarian medusae, recent molecular studies indicate that the ctenophores are only distantly related to the cnidarians, which together with the bilateria constitute the eumetazoa, true animals. When we think of animals, we usually think of eumetazoa, since most animals fall into this category. Link to learning. Watch a presentation, http://openstax.org/l/saving_life, by biologist E. O. Wilson on the importance of diversity. Animal reproduction and development. Most animals are diploid organisms, meaning that their body, somatic, cells are diploid and haploid. Reproductive, gamete, cells are produced through meiosis. Some exceptions exist. For example, in bees, wasps, and ants, the male is haploid because it develops from unfertilized eggs. Most animals undergo sexual reproduction. However, a few groups, such as cnidarians, flatworms, and roundworms, may also undergo asexual reproduction, in which offspring originate from part of the parental body. Processes of animal reproduction and embryonic development. During sexual reproduction, the haploid gametes of the male and female individuals of a species combine in a process called fertilization. Typically, both male and female gametes are required. The small, motile male sperm fertilizes the typically much larger, sessile female egg. This process produces a diploid fertilized egg called a zygote. Some animal species, including sea stars and sea anemones, are capable of asexual reproduction. The most common forms of asexual reproduction for stationary aquatic animals include budding and fragmentation, where part of a parent individual can separate and grow into a new individual. This type of asexual reproduction produces genetically identical offspring, which would appear to be disadvantageous from the perspective of evolutionary adaptability, simply because of the potential buildup of deleterious mutations. In contrast, a form of uniparental reproduction found in some insects and a few vertebrates is called parthenogenesis, or virgin beginning. In this case, progeny develop from a gamete, but without fertilization. Because of the nutrients stored in eggs, only females produce parthenogenetic offspring. In some insects, unfertilized eggs develop into new male offspring. This type of sex determination is called haplodiploidy, since females are diploid, with both maternal and paternal chromosomes, and males are haploid, with only maternal chromosomes. A few vertebrates, e.g., some fish, turkeys, rattlesnakes, and whiptail lizards, are also capable of parthenogenesis. In the case of turkeys and rattlesnakes, parthenogenetically reproducing females also produce only male offspring, but not because the males are haploid. In birds and rattlesnakes, the female is the heterogametic, ZW, sex, so the only surviving progeny of post-meiotic parthenogenesis would be ZZ males. In the whiptail lizards, on the other hand, only female progeny are produced by parthenogenesis. These animals may not be identical to their parent, although they have only maternal chromosomes. However, for animals that are limited in their access to mates, uniparental reproduction can ensure genetic propagation. In animals, the zygote progresses through a series of developmental stages, during which primary germ layers, ectoderm, endoderm, and mesoderm, are established and reorganized to form an embryo. During this process, animal tissues begin to specialize and organize into organs and organ systems, determining their future morphology and physiology. Animal development begins with cleavage, a series of meiotic cell divisions, of the zygote, figure 27.3. Cleavage differs from somatic cell division in that the egg is subdivided by successive cleavages into smaller and smaller cells, with no actual cell growth. The cells resulting from subdivision of the material of the egg in this way are called blastomeres. 3 cell divisions transform the single-celled zygote into an 8-celled structure. After further cell division and rearrangement of existing cells, a solid morolla is formed, followed by a hollow structure called a blastula. The blastula is hollow only in invertebrates whose eggs 27.1, features of the animal kingdom 701 have relatively small amounts of yolk. In very yolky eggs of vertebrates, the yolk remains undivided, with most cells forming an embryonic layer on the surface of the yolk, imagine a chicken embryo, growing over the egg's yolk, which serve as food for the developing embryo. Further cell division and cellular rearrangement leads to a process called gastrulation. Gastrulation results in 2 important events: the formation of the primitive gut, archenteron, or digestive cavity, and the formation of the embryonic germ layers, as we have discussed above. These germ layers are programmed to develop into certain tissue types, organs, and organ systems during a process called organogenesis. Diploblastic organisms have 2 germ layers, endoderm and ectoderm. Endoderm forms the wall of the digestive tract, and ectoderm covers the surface of the animal. In triploblastic animals, a 3rd layer forms, mesoderm, which differentiates into various structures between the ectoderm and endoderm, including the lining of the body cavity. Figure 27.3 Development of a simple embryo. During embryonic development, the zygote undergoes a series of meiotic cell divisions, or cleavages, that subdivide the egg into smaller and smaller blastomeres. Note that the 8-cell stage and the blastula are about the same size as the original zygote. In many invertebrates, the blastula consists of a single layer of cells around a hollow space. During a process called gastrulation, the cells from the blastula move inward on one side to form an inner cavity. This inner cavity becomes the primitive gut, archenteron, of the gastrula, little gut, stage. The opening into this cavity is called the blastopore, and in some invertebrates it is destined to form the mouth. Some animals produce larval forms that are different from the adult. In insects with incomplete metamorphosis, such as grasshoppers, the young resemble wingless adults, but gradually produce larger and larger wing buds during successive molts, until finally producing functional wings and sex organs during the last molt. Other animals, such as some insects and echinoderms, undergo complete metamorphosis in which the embryo develops into 1 or more feeding larval stages that may differ greatly in structure and function from the adult, figure 27.4. The adult body then develops from 1 or more regions of larval tissue. For animals with complete metamorphosis, the larva and the adult may have different diets, limiting competition for food between them. Regardless of whether a species undergoes complete or incomplete metamorphosis, the series of developmental stages of the embryo remains largely the same for most members of the animal kingdom. 702 Chapter 27, Introduction to Animal Diversity. Access for free at openstax.org. Figure 27.4 Insect metamorphosis. A. The grasshopper undergoes incomplete metamorphosis. B. The butterfly undergoes complete metamorphosis. Credit. S.E. Snodgrass, USDA. Link to learning. Watch the following video. http://openstax.org/l/embryo_evol to see how human embryonic development, after the blastula and gastrula stages of development, reflects evolution. The role of homeobox, HOX, genes in animal development. Since the early 19th century, scientists have observed that many animals, from the very simple to the complex, shared similar embryonic morphology and development. Surprisingly, a human embryo and a frog embryo, at a certain stage of embryonic development, look remarkably alike. For a long time, scientists did not understand why so many animal species looked similar during embryonic development but were very different as adults. They wondered what dictated the developmental direction that a fly, mouse, frog, or human embryo would take. Near the end of the 20th century, a particular class of genes was discovered that had this very job. These genes that determine animal structure are called homeotic genes, and they contain DNA sequences called homeoboxes. Genes with homeoboxes encode protein transcription factors. One group of animal genes containing homeobox sequences is specifically referred to as HOX genes. This cluster of genes is responsible for determining the general body plan, such as the number of body segments of an animal, the number and placement of appendages, and animal head-tail directionality. The first HOX genes to be sequenced were those from the fruit fly, Drosophila melanogaster. A single HOX mutation in the fruit fly can result in an extra pair of wings or even legs growing from the head in place of antennae. This is because antennae and legs are embryologic homologous structures, and their appearance as antennae or legs is dictated by their origination within specific body segments of the head and thorax during development. Now, HOX genes are known from virtually all other animals as well. While there are a great many genes that play roles in the morphological development of an animal, including other homeobox-containing genes, what makes HOX genes so powerful is that they serve as master control genes that can turn on or off large numbers of other genes. HOX genes do this by encoding transcription factors that control the expression of numerous other genes. HOX genes are homologous across the animal kingdom. That is, the genetic sequences of HOX genes and their positions on 27.1 features of the animal kingdom's 703 chromosomes are remarkably similar across most animals because of their presence in a common ancestor, from worms to flies, mice, and humans. Figure 27.5. In addition, the order of the genes reflects the anterior-posterior axis of the animal's body. One of the contributions to increased animal body complexity is that HOX genes have undergone at least 2 and perhaps as many as 4 duplication events during animal evolution, with the additional genes allowing for more complex body types to evolve. All vertebrates have 4, or more, sets of HOX genes, while invertebrates have only 1 set. Visual connection. Figure 27.5 HOX genes. HOX genes are highly conserved genes encoding transcription factors that determine the course of embryonic development in animals. In vertebrates, the genes have been duplicated into 4 clusters on different chromosomes: HOX A, HOX B, HOX C, and HOX D. Genes within these clusters are expressed in certain body segments at certain stages of development. Shown here is the homology between HOX genes in mice and humans. Note how HOX gene expression, as indicated with orange, pink, blue, and green shading, occurs in the same body segments in both the mouse and the human. While at least 1 copy of each HOX gene is present in humans and other vertebrates, some HOX genes are missing in some chromosomal sets. If a HOX13 gene in a mouse was replaced with a HOX1 gene, how might this alter animal development? 2 of the 5 clades within the animal kingdom do not have HOX genes: the Ctenophora and the Porifera. In spite of the superficial similarities between the Cnidaria and the Ctenophora, the Cnidaria have a number of HOX genes, but the Ctenophora have none. The absence of HOX genes from the ctenophores has led to the suggestion that they might be basal animals, in spite of their tissue differentiation. Ironically, the Pachyzoa, which have only a few cell types, do have at least 1 HOX gene. The presence of a HOX gene in the Pachyzoa, in addition to similarities in the genomic organization of the Pachyzoa, Cnidaria and Bilateria, has led to the inclusion of the 3 groups in a parahoxozoa clade. However, we should note that at this time the reclassification of the animal kingdom is still tentative and requires much more study.